Thank you. And first of all, I really thank the organizers for inviting me. It's been a wonderful meeting. I've had a great time. I feel like I used to feel when I would come to meetings in Antwerp many years ago, so thank you. So we're interested in myelin proteins and how they cause Charcot-Marie tooth disease, and we've spent a lot of time over the years looking at myelin protein 0 and CMT1B. But we've recently found that P0 interacts with PMP22 directly, and we've seen CMT1E patients over the years, so we elected to devote our attention to CMT1E, which is caused by mutations in PMP22, though not a duplication of PMP22. And on the left here, I think you see a two-dimensional image of the protein. It's a tetramembrane protein, and we wanted to look again at the phenotypes of patients with CMT1E, so we screened the Inherited Neuropathy Consortium databases, and we found 50 patients with 24 different mutations. And we were sort of surprised to find that almost all the mutations occurred within the transmembrane domains. And in the middle, you see an AlphaFold model of PMP22, and you can see that a lot of the mutations are in the green band coming down there, which is transmembrane domain 2, which we were also interested in because transmembrane domain 2 is the portion of PMP22 that interacts with P0. And this is really just to show that a lot of these patients had significant pathology at a young age. These are just some of the standard ways we look at patients, which I think many of you are familiar with. And again, the different mutations here you can see on the bottom, and these are all in the transmembrane domains. So we wanted to start by just looking at how these different variants progress in our patients. And so I'm just going to show you initially just one mutation, which is the leucine 82 proline, just to show you that on the x-axis is age, on the y is a CMT exam score, and just to show that these progress actually very quickly over the first few years from mild into a severe range, and this is true for many of them. And then when we put all of this together, it's a busy slide, but what I want to try to point out here is that, first of all, again, the age is on the bottom, and many of the patients with the transmembrane domain variants progress within the first 2 decades of life. But a person could have the same score at age 10 that somebody else might have at age 50, and we wanted to be able to sort of put this into perspective, to be able to sort of get a better model of severity. So what we developed was what we call a CMT progression index. And this we were able to do with patients that we'd seen more than one time over the years. So we're able to sort of get a sort of an S-shaped curve, which you can see there, which would sort of predict how patients might progress if we saw them with particular scores at a couple of time points. And the progression index works from 1 to 10, and 10 is the most severe. And then we knew from the literature, going back with Jim Lepsky's group and others, that the Trembler J mutation, which is the L16P, was retained in the endoplasmic reticulum. And these are, on the right is images from our group where we'd use permanent transfections. And PMP22 is labeled in green. And on the top in the wild type, you can see it makes it out to the cell surface. These are in HEC293 cells. And you can see that it's retained in the ER with the Trembler J mutation. And we knew of the work with unfolded protein response with many people, and I have to give a shout out to Larry Rabbits, who was one of the first to really develop this concept. And so we wanted to be able to look to see what was happening with ER retention with our PMP22 variants. So here on the left is an AlphaFold model showing the progression index. And you can see the deeper you get into the membrane, the higher and more severe the progression index is. And then we were able to use a cell surface labeling model. And on the right, you can see the different mutations. And if they go out to 100, it means they're getting out to the cell surface. And you can see that many of the mutations, I'm sorry, I don't have a pointer, but you can see the Tremblor J mutation, which is L16 Pro, and many others really are near the 0 point on the x-axis, really suggesting that these are retained in the ER. And we wanted to see how that correlated with our progression index. And again, the progression index of 10 is the most severe, and you can see that the severity really correlates quite well with the inability of the mutated protein to get out to the cell surface. And again, these are in HEC293 cells. So where we're going with this is we want to move forward to really try to get an understanding of the pathogenesis, and using our cell lines, really look at the UPR, but also look at other potential mechanisms that can affect the biology of the cell that can contribute to the disease, such as what's happening with the wild-type protein, how these proteins are being degraded. And then we plan to make mouse models looking at several of the different pathogenic mechanisms we see, and then to provide or develop a rescue mouse to sort of see what happens when we genetically eliminate the cause of the disease at different ages in the mice. And I'll spend the rest of the talk just briefly showing you a few examples of that. So first of all, I just want to show the value of having the permanent versus the transient transections. So on the immunofluorescence image, this is wild-type PMP22, and when you transiently transfect these HEC293 cells, you can see that you really get a lot of the protein still hung up in the ER. But when you have the permanently transfected cells, you can see that really on the wild-type, it's all getting out to the cell membrane. And so, again, all of the cells we're using are permanently transfected, and we plan to use these to look at the UPR in standard assessments. And then we're also interested in what's happening because of the mutant with the wild-type allele. So is there sort of a dominant negative effect? And you can see this in sort of the cartoon in the middle. We want to see if the mutant protein really prevents the wild-type protein from getting out to the cell surface. And I don't have a pointer, but on my left, those are immunoprecipitation assays, which shows that with the 3 examples of mutant PMP22 introduced into the system, we immunoprecipitate the wild type as well. And on the right, it shows, again, it's hard to, without a pointer, but we have dual labeled constructs, the wild type with the blue and the mutant with the red. And you can, when you do fluorescent tagging of the cell surface, you can see that actually the wild type is being held down by the mutant. And then the last thing we wanted to look at was really degradative mechanisms, because as I'll show you on the next slide, some of these mutations, actually you can see like the L16P and the S76I, not only is there retention within the ER, but there's really aggregates forming, suggesting that there's autophagy or autophagy pathways that we want to look at. Yet on other severely affected patients like the L147R, you see a totally different distribution in the ER, although there's still ER retention. So our goal there is to develop mouse models with these mutations to try to look at potential different mechanisms contributing to the disease. On the left is a mouse model that we created. There's a film clip, but it keeps crashing every time I try to put it into the talk. So you have to take my word that that shows a mouse who is severely disabled. And again, that's with an 11 amino acid deletion in the second transmembrane domain. And you can see that the phenotype in these cells is very similar to what we see in L147, but different from what we see in S76I. Then the last thing that we want to do is we want to develop a rescue animal where we genetically use a CRELOX system to basically take out the mutation at different ages using CRE, so that we can, with this mouse, we can like sort of look at different ages of potential intervention and see what happens when we try to treat an animal early in life or later in the disease. And then this also would give us a model to look at biomarkers before and after treatment to try to develop, to determine which markers would be treatment responsive, and again to look at drug-specific and gene-specific effects. And getting to Steve's point from a little earlier, we're going to be looking by morphology, electrophysiology, and also by behavior. So that is sort of our plan. This is our team. On the left is the group, our clinical research group at Iowa. In the middle is the Piper Lab, also at Iowa. And then, of course, we couldn't do any of our work without help from patients, and that's what's shown on the right. And I thank you for your attention.
Thank you, Mike, for your nice talk. There's room for 2 quick questions from the audience. Mary?
I wasn't going to answer any further questions.
Hi, Mike. Lovely talk. Very interesting.
Thank you.
Can I just double check? So in terms of the 2 mechanisms,
Yes.
do you think the cause of both the mutated allele and obviously the wild-type allele not getting to the plasma membrane is the dominant negative effect of the interaction? Or do you think it's intrinsic to the mutation that it won't get there? Or a bit of both?
Well, so I'm going to answer the question the way I understand it, which is not exactly
Sorry.
phrased. No, because I think there's 2 possibilities. One is if there's activation of the unfolded protein response. Part of that is caused by sort of stress in the UPR. And if the mutated protein is bringing the wild type down also, it's just more work for folding for the ER. So I think that's part of it. But also, if it can't get out to the cell surface, then it can't really interact with P0, for example. So I think that can be contributing to the disease also. Does that answer your question? Or?
Thank you.
Okay.
Any other question? Oh,
I think
Vincent,
Vincent.
and then we move.
So what about chaperoning these mutations? So calnexin is a chaperone of PMP22.
I'm sorry, I didn't understand the question.
The chaperones that can refold
Yes,
this
yeah.
mutated protein may also play a role.
I
in
agree
your
with that.
story.
Yeah.
Like calnexin is a known
Yeah.
chaperone of
I-
PMP22, but also the icho proteins might be involved there.
I agree with that, Vincent, and that's something we have to find out.